So we had a video where we talked about the chemistry that can happen between amino acids, whether it's the amino and the acid functional group that do chemistry to generate the peptide bond that links those amino acid monomers together. We're starting to think about the chemistry that can happen between side chains like the thiols and cysteine to create disulfide links. We also have acid-base properties to think about with our amino acids. So before we actually think about amino acids, let's take a second and just review here with what we know and thought about with monoprotic acids, something like acetic acid. Here we have the first deprotonation of phosphoric acid and then ammonium ions. So all of these are monoprotic, meaning that there is one proton to remove. Now I'll highlight here, if we're talking about phosphoric acid, there really are 3 protons total, but we're just considering this second proton that's coming off here with phosphoric acid. But when we have monoprotic acids, this titration curve, if you will. Remember, this is really showing a change in pH as a function of the number of protons that are dissociated from a population of molecules. And we highlighted that there's something special that happens at this halfway point. So this halfway point represents the pKa of whatever acid that we're talking about. And that halfway point is really important because we could define pKa also by saying that it is the pH to be at for half of your molecules to be without their proton. So that was a way to sort of define pKa. We could say that pKa is defined as the pH for you to be at for half of your molecules to be without their proton. I'm just going to give myself a little bit more room here for the next piece here. Okay, so that was just a review and just highlighting there's lots to do with the importance of halfway values in biochemistry. Okay, so what's different then when we think about amino acids? So let's highlight here, let's draw a structure of amino acid for a second and remind ourselves of the relevant parts. So an amino acid always has an amine group and then a carboxylic acid group. There's always a hydrogen atom on that alpha carbon and then a side chain. So when we think about pKa values, right? The pKa value of a protonated amine is 9 to 10. And that's important because that reminds us that that functional group is going to be protonated at physiological pH because pKa is greater than pH. Conversely, when we think about the acid functional group, okay, pKa here is going to be equal to 3 to 5. And so for that functional group, we're going to be deprotonated because pH is greater than pKa. So we have these Zwitter ions, molecules that have no net charge, but still are not uncharged. Okay, so you're never going to see an amino acid that has, not talking to Siri there, you're never going to see an amino acid that has a charge that is uncharged. You're never going to have an NH2 group and then a COOH group there. Okay, so here's sort of our generic amino acid. So we really can think of amino acids as being at minimum diprotic acids. And that's because of those 2 ionizable functional groups. Now, you potentially have the ability of having an ionizable group here in your R group. So your R group also has the ability to have an ionizable group. And once you start learning your amino acids, you'll remember, oh wow, there's 2 that have carboxylic acid functional groups. There's 3 that have protonated amine functional groups. So there's actually a number of amino acids that can contribute additionally to sort of this acid piece. But let's just go through, let's take the simplest case where R is equal to hydrogen. And so let's look at glycine. So what we have here is our first pKa value in glycine here corresponds to that carboxylic acid group losing its proton and becoming a carboxylate group. And we know, right, that we're going to have that carboxylic acid pKa value of around 3 to 5. Okay, now it's actually a little bit lower than 3 here, and we talked about the reason for that is if you actually have a positive charge that's over here that's going to help pull away electron density, and so that's actually going to make that carboxylic acid more acidic, decreasing its pKa value. Okay, on the flip side, when we think about what happens with this second pKa value, that's corresponding to the deprotonation now of our protonated amine. And reminding ourselves that that protonated amine has a pKa of around 9 to 10, okay? And one of the things I want to go through and sort of highlight here is when we're all the way down in the acidic spectrum, we're going to end up with a molecule that has a +1 charge. And then once we lose that proton on that carboxylic acid, we end up with that Zwitter ionic species that now has no net charge. It's not that it's uncharged, it has no net charge. We're going to be continuing to increase the pH, and it's not until we get to close to the pKa value for that protonated amine that we begin to start deprotonating again. And so again, we'll go from a species that has no net charge to now having a species that has a -1 charge. So when we were going through sort of these transition regions, this halfway point corresponds to the pKa for that corresponding functional group. Same thing here, pKa for that functional group. So what we're going to see now is there's a special point that's halfway between the halfway points. And that's this special point that we have right here. This special point is called a pi value. We're going to talk about sort of what that is here. So we can define and think about pi like being the pKa for a protein. So imagine a protein here, right? Proteins really are going to be hooking together all of these amino acids. So even though each amino acid comes with a +1 and a -1 group, once we couple them together, only the one on the far left is going to have a +1 charge. Everything in the middle is not going to have a charge on the backbone, and only the one on the end is going to have a -1 charge on the carboxylate group. So we're going to have a +1 on the N terminus, a -1 on the C terminus, and then potentially whatever dangles, whatever R groups we have that have ionizable side chains, we potentially have those contributing to what the overall charge is. So just like for a molecule, we could think about pKa as being the pH to have half of your molecules be without their protons. We can define pi like being a pKa for a protein as being the pH at which your molecule, which in this case is going to be a protein, has no net charge. So pi is going to be the pH at which your molecule, and we're specifically going to be talking about proteins here, has no net charge. So this is a really important property for proteins because as we're going to see when we study things in lab, when your protein has no net charge, note that we're not saying our protein is uncharged, but when all the pluses is balanced, all the minuses, and we have no net charge, that is going to give a protein a particular property, especially when we're talking about how it behaves in an electrophoresis field. It's not going to move. And so we can do really unique things with electrophoresis with proteins if we are at the pH that matches the pi of a protein. So we'll get to that in lab. But key here is to recognize that when we're thinking about acid-base properties of our amino acids and then our proteins, we've got multiple ionizable groups to think about. So to simplify that, we define essentially the pKa of that protein as being something that we call the pi. Pi of a protein is the pH you want to be at where your protein has no net charge.